Let's begin with prayer. Heavenly Father, thank you. Thank you for the light that you've given us in Jesus. Thank you for your Holy Spirit that gives our minds the ability to understand, and that you guide us into all truth. Father, I pray you might be pleased today, that you might help us understand these things, help us be encouraged that your word is true from the very first verse, and that we can rely upon it. And so I pray that we all might be encouraged, come away encouraged today. We just want to commit this time to you for your pleasure, for your glory. Thank you. In Jesus' name. Amen.
Amen.
Okay, last week we got a start on these 10 huge reasons why Darwinism is not viable as a scientific idea. We actually did not get through even the first point. So, but let me just explain. Let me explain that first session was laying a lot of groundwork, helping you have enough background in biology to understand the other, not only the first point, but all 10 points, because they're all connected. They're all very much built upon what we've discovered in the biological world, especially in the last 60 years. So once we get through this first point, I expect to move much more quickly. And I am still hoping to finish in 4 weeks. And one other thing is that we're planning to take a break on Easter, not have a lecture. So we'll meet, Lord willing, the week after Easter. So, okay. Again, I'll put up the slide I had last week. Why should we as Christians even care whether or not Darwinism is viable as a scientific idea? And again, it's because Darwin's ideas are an essential part of a hostile worldview which is in bitter conflict against God and against us as Christians in our culture today. It's taught as dogma in our schools, from elementary school through the university. We're blasted with it in the media. Just every direction we're blasted with this perspective that is very hostile to God and hostile to the Bible. And to me it's very clear Satan is using Darwin's ideas to deceive multitudes around us, including in many cases our own children and grandchildren. So this is, if we are, especially, as parents, we have accountability to God for our children. We have love for our children. We desperately don't want them to fall prey to this deception. So that should be motivation enough for us to seek to be well equipped, understand what the answers are, and be ready to defend biblical truth. Last week we saw, I just tried to highlight the contrast between these two worldviews. One summarized in the very first verse of the Bible, in the beginning God created the heavens and the earth, versus a foundational statement by Carl Sagan in the Cosmos series back in the 80s, where Sagan says, the cosmos is all that is or was or ever will be. So that epitomizes this other worldview, this materialist or naturalistic or atheistic worldview. And last week, in way of introduction, I gave a little background on Darwin, and that he was definitely a product of the Enlightenment. His grandfather was very active in Enlightenment circles in England. And that his grandfather actually, in a book he wrote, actually laid out the basics of the theory that Darwin published some 70 years later. So these ideas come out of this what I call last week a radical atheist movement called the Enlightenment. And it has profoundly affected our world even today. Now, I pointed out that Darwin got his basic ideas from considering the success of plant and animal breeders of his day. And many of the breeds of cattle that we have were developed right there in England. The Jersey, the Hereford, and the Angus are English breeds of cattle. And they are specifically selected by using artificial selection, human selection, to get very beef animals that are very good for beef production, and dairy animals that were did very well as far as milk production and butterfat production. So there's no doubt that these methods of selection work. You see in the various dog breeds, all these different breeds, and most of these were bred for specific purposes. Many of these breeds are work dogs, used for herding sheep, or guard dogs, or retrievers, and so on. So basically these breeds, basically Darwin didn't understand it, and most evolutionists don't understand it. They're basically utilizing the genetic diversity that God built into the created kinds when He created the world and created the life upon it, created human beings, and this diversity is basically all good. And what it does is enables various types of plants and animals to adapt readily and quickly to a given ecological niche. And so that's part of God's design, part of God's plan. And so, but okay, so the essence of Darwin's theory was that, analogous to the manner in which plant and animal breeders developed improved varieties, Darwin claimed that natural processes could lead to types of plants and animals better suited to the environments in which they find themselves. How does this occur? Well, variations in the physical characteristics of a given plant or animal type give some individuals an advantage over others. Individuals with the advantage tend to leave more offspring than the others. If the trait responsible for the advantage is heritable, the offspring will tend to have the trait and also display the advantage. If enough generations elapse and environmental conditions don't change, eventually most individuals in the population will have that beneficial trait. And that tendency is accentuated by the fact that most organisms produce many more offspring than can be supported by the resources available. Hence, in every generation, there is usually a competition or struggle, and often a fierce one, for survival. And so Darwin described this process as natural selection as opposed to artificial selection that's practiced by plant and animal breeders. And so I asked, well, does this really happen? Does this really occur? And to me, it's obvious. The answer is yes. However, the more important question is just what sort of changes in populations is natural selection capable of producing? Is it capable of unlimited change, unlimited modification, or are there strict limits? And I think I said last week, though, it's been discovered in plant and animal breeding that there are hard limits to how much change is possible. It's been found that, for example, sugar beets, only a certain maximum amount sugar content of sugar beets was possible through selective breeding. Only a certain maximum level of butterfat production was possible in the breeding of dairy animals. And so, contrary to Darwin's belief, there are strict limits to how much variation is possible, at least in the variation that God produced in the various created kinds. So that was a big problem. Darwin admitted he did not know how this variation, where this variation came from, how it appeared. he was ignorant. He was ignorant of the laws of heredity, and so had he known what is known today, chances are he would not have proposed his theory. So a lot of his optimism, a lot of his confidence was basically based in ignorance. And some of the things that Darwin did not understand, one of the biggest thing was how much his theory would ultimately need to explain. He had no inkling of the complexity of life at the molecular level, what has been discovered over the last 60 years. His understanding of the complexity of life was limited to what an optical microscope could reveal. And the resolution of an optical microscope is limited to a single wavelength of light. An optical microscope cannot show any features that are smaller than about a half a micron, which is the wavelength in the middle of the optical spectrum. So Darwin was ignorant of the finer details, even in a living cell. He basically couldn't distinguish much more than the cell nucleus in the living cell. And so he was ignorant of the complexity. He was ignorant of, as I've already said, about where the variations that plant and animal breeders use, where these variations come from. He primarily thought it had to do with changes in environmental conditions. He thought simply by changing environmental conditions, these new characteristics just spontaneously appeared. And with that belief, he thought that infinite amount of variation could potentially arise. But that we know now it was an error. A third point was he had no inkling that the specifications that describe the characteristics, physical characteristics of living organisms are in the form of coded linguistic instructions. And we'll hopefully get to that today, the significance of that. And finally, he was ignorant of the laws of inheritance, which his contemporary Gregor Mendel in Austria discovered, but Mendel's publications were not widely disseminated, and as far as we know, Darwin never knew about them during his lifetime, and they really didn't become widely known until 1900. But after the turn of the 20th century, there were quite a few discoveries made. I'd say one of the discoveries was the discovery of radioactivity and the fact that radioactivity could cause mutations. And so about the time in the early 40s, a revised version of Darwin's theory was published, was accepted generally, and that revised version today is known as Neo-Darwinism. And its main features are, it contains Darwin's basic idea of natural selection. It has folded into it Mendel's laws of inheritance, which basically said that the elements of inheritance are discrete, and they're passed from parents to children in discrete units we know as genes. Okay, so that's the main aspect of what Gregor Mendel discovered, and that some of these traits are dominant, some are recessive. That's part of it as well. And the third item, a fairly big part of it, is that the source of the variation is understood to be random genetic mutations of the sort that radiation produces. So that's understood to be the source of the variation. Nothing about the variations that God put into the organisms originally. So a neo-Darwinist has to rely on random, basically the overwhelming majority of these mutations destroy genetic information, are destructive and bad, and lead to bad consequences. But anyway, that's all a neo-Darwinist has to work with, those random bad mutations. Okay, so this is the theory that I'm critiquing today, neo-Darwinism. But certainly prominent in neo-Darwinism are Darwin's ideas, namely natural selection. Okay, and so we're looking at 10 major reasons why neo-Darwinism is bankrupt now as a scientific idea. And I have provided you a list of these 10 items. I have them here. First, life at the molecular level is unimaginably complex, almost beyond the human mind to even grasp how complex it is. But beyond that, it is what I'm terming irreducibly complex, and I'll explain what that means. Second, life specifications are non-material, and therefore beyond the reach of a material explanation like neo-Darwinism. Neo-Darwinism is an attempt to explain how life arose purely in terms of physical processes that we can study in chemistry and physics. And what this second point says is that the even more important aspect of living things is that it's based on non-material reality. 3, the cost of substituting a new trait for an old one by natural selection is extremely costly. It is very high. And this puts the time span required for anything significant to change far beyond the reach of neo-Darwinism. So this third point says that just the trying to replace an old trait with a new trait takes a long time, many generations, and a large number of people, of individuals reproducing that they can't reproduce if that selection process is to take place. So that means most people's offspring can't leave other offspring. Fourth, natural selection cannot even detect, it's been discovered, most bad mutations, and hence cannot prevent genetic decline and extinction. So rather than this process of mutation and selection leading to improvement, it leads to just the opposite. It leads to genetic decline. Another gigantic reason neo-Darwinism doesn't work. Okay? Fifth, favorable mutations are so rare as to be non-existent relative to deleterious mutations. And this is a consequence of the way this genetic information is encoded in DNA. There are many overlapping messages. A single genetic letter is part of multiple overlapping messages. So if you change one letter, and it might be neutral in one message, or perhaps could conceivably give a slight benefit in one of the messages, it almost certainly is destructive in all the other messages. 6, even if favorable mutations do occur, the waiting time for even short sequences of them, just a sequence of 2, for them to arise is prohibitively long. And so that has been demonstrated, and I'll talk about that. 7, the prevalence of orphan genes shows that Darwin's concept of common ancestry can no longer be defended, because it's been found that essentially every species, certainly every genus, has genes that are unique to that genus. They can't be traced to any ancestral form. So that's clear evidence of God creating each type supernaturally independently, so that you can't produce a ancestral tree like Darwin published in his book. 8, staggering number of transitional forms that Darwin's idea, this gradualistic process requires, are systematically missing from the fossil record. They're just not there. Darwin acknowledged that, but he had faith that sometime they would be discovered, and they haven't been. 9, Darwin's idea of increasing diversity of life with time is falsified, again by the fossil record, by the fact that at the very beginning of point in the record where you find multicellular life, essentially all animal phyla appear abruptly. Something like 23 of the main animal phyla are there in the very lowest fossil-bearing rocks. So that turns Darwin's scenario on its head. And 10, radioisotope evidence shows that the Earth is only thousands of years old, not billions, so that the vast time span that Darwin envisioned simply does not exist. And that's probably hard for, we've been so bombarded with the idea that the Earth is ancient. There's probably some doubt in most of your minds that there's real evidence to back this up, but I plan to provide that evidence, Lord willing. Okay, last week we began our consideration of the first item. Life at the molecular level is unimaginably complex, and we basically almost got through that. So, and I had a quote from Darwin's. From this book, Darwin's Black Box by Michael Behe. And I recommend this book if some of you want some additional reading. In his introduction to the book, Behe says, modern science has learned that ultimately life is a molecular phenomenon. All organisms are made of molecules that act as the nuts and bolts, gears and pulleys of biological systems. And that really hardly captures the reality of it. That our cells are just packed with little machines that have moving parts, and that are able to walk down the skeleton of the cells, carrying containers of various chemicals from one point to the other. And there's just incredible motion and activity and machine-like behavior going on in our cells. And this is, I showed this last week, animation of a biped motor protein known as kinesin walking along a microtubule. So it's the part that's walking, that's a single protein machine that is able to do that. And like I said, there's, in addition, on these little containers that these things carry, there are addresses, like a UPS label to tell that machine where to take it. Just unbelievable complexity. This is another rendition of that same machine carrying the transport vesicle. And then we spent quite a bit of time looking at another example of these machines in living cells. And this particular machine is called ATP synthase. And it basically recycles a molecule called ADP, adenosine diphosphate, into the energy-rich molecule ATP, or adenosine triphosphate. And many of our cells have hundreds of thousands of these machines in a single cell. They spin at about up to about 10,000 RPM, and they are responsible for recycling approximately our body weight worth of ATP from ADP back into ATP. That say if you weigh 150 pounds, your body recycles about 150 pounds worth per day of that material. And even though you only have about 1 to 2 ounces of ATP in your body, it gets turned over about once a minute. And so that adds up to about your body weight over a course of a day. And so there's these and it turns out, or here's the ATP molecule. And it essentially, all the processes in our body, most of these little machines use this molecule to fuel to give it its the power it needs. That little biped robot needs one molecule of ATP for each step it takes. Okay, so these processes, these cellular processes, use a lot of this material, and the way the energy is released is when this phosphate unit here breaks away. There's just about a nice amount of energy released that empowers these cellular processes. And so what ATP synthase does is join that phosphate back to here using energy from the environment, energy that are produced from the food that we eat. It enables it to put it back together and recycle that ATP. Okay, and so I showed this another version, another schematic of this motor, of this machine, showing and emphasizing here there are about 40,000 atoms. So this is a complex machine, and every atom makes a difference. If you start getting some of the atoms wrong, the machine doesn't work, or if it does, it works less efficiently. So anyway, this part right here rotates, and the ADP comes into here plus phosphate, and they get crunched together. There's catalytic activity in this part, so that reaction is catalyzed as well as mechanically they get pressed together, and then they come out as ATP. So that's what's going on. And as I said, these little machines are in every form of life on Earth: bacteria, plants, and animals, and humans. So every form of life uses this energy mechanism and this way to reconstitute ATP. And this is a little animation. And the purple little things are the ATP, and the green are the ADP going in here and getting scrunched together. And this is the rotation that drives this scrunching together. And this rotation is driven, it's essentially an electrical motor. Protons are going through here. They're higher in concentration on the outside of the membrane than the inside, and that's what drives that machine. So it amounts to not only a rotary machine, but a electrically driven, like a motor, like an electric motor. And just imagine this going on, and us having, in many cases, in our muscles and our liver and our heart muscles, there can be 1,000 mitochondria, the envelopes that contain these, and as many as 1,000 in each mitochondria. Yes?
Are these processes that you're describing observable or theoretical?
They are, well, first of all, the size of them it's extremely tiny. There was a breakthrough as far as the way we got more resolution beyond what an optical microscope could provide. In the 1930s, what's called an electron microscope was invented in Germany. And so it uses electrons to visualize the structure. And so as you increase the energy level, you can resolve, of the electrons, you can resolve finer and finer structures. With the electron microscope, they were able to see these, to image these mitochondria that contain these. And even it's possible to image at least the rough details of these machines. They had to resort to other kinds of techniques to actually work out the structure. That this structure was worked out in the 1990s. The 1997 Nobel Prize in biology was awarded to the guys that worked out this figured out this structure. So and you know, to test the rotation rate, it's been possible to put a little mirror, if you will, on this rotating part of it and actually measure the rotation rate. So they use incredible technology to work out the structure here. Yes.
So would you say that any breakdown in that would be the beginning of disease?
Well, there are, and as I said last week, one aspect of aging is the fact that these the specifications for making these machines start to have errors, mutations, and so these mutations are not made to spec, or those specs are degraded, and so the machines don't work as well, and so that's one reason we don't have the energy that we had when we were younger, is that our ATP synthase machines are not working at the efficiency they once worked at. So, that's I mentioned last week this product that Mary sells.
Oh, yeah.
Ovation.
And it you know, it's based on research with laboratory animals. They advertise it to make you live 25% longer and feel 25 years younger.
Is that why you don't look your real age?
And as I joke, that's how I'm able to. That's why at 87 years old I'm able to.
Oh, doesn't this support the materialist perspective that they think that everything can be explained because you know all these things, you know these machines work, you know what they do, and so that's why they think everything's material.
Right. So this they think that just because they have worked out these details, that their intellectual framework is valid. But you know, they have no way, as I'll show, they have absolutely no way of explaining how such a thing could have originated apart from God by any kind of conceivable material process. And we're going to see why it's just completely irrational to think a material process could give rise to something like that.
When I studied these in college, the traditional explanation of how they evolved was that, if you go back to the picture, it's very unsatisfying. The traditional explanation that I learned was that in college was that the part on the bottom functioned by itself and the part on the top functioned by itself independently, and then all of a sudden they just sort of clicked together and ended up working better.
Yeah.
That's the explanation.
Yeah. But because this provides the energy for essentially every kind of cellular process, including the translation of RNA, the instructions into proteins.
You can't get there from here.
That's absurd.
Yeah, exactly.
That's dishonest to put out something like that.
Yeah, there's no good. You're right. There's no good explanation.
Yeah.
I just find it fascinating that anything that man has thought up on a blueprint or has come up with in a motor, that God already had the template and he has allowed us to think it up, but he already made it. I mean, Adam's first breath, there was electricity in his heart. And then it took us centuries to come up with electricity. It's pretty amazing.
Yeah, I mean the technology and the design and it so far surpasses what we as humans have been able to do. I mean, even the technology in our cell phones is crude, child's play compared with what the kind of technology we're looking at here. Okay, so I want to take this to the sort of the next level and point out that all this complex machinery we just looked at has to be manufactured within the cell. So just what kind of technology is involved in making machines like this? So that's what we're gonna look at next. And so the detailed specifications for these machines is those are encoded within an organism's DNA, and so which is this helical double helix molecule. I'm sure you've all heard about that, and it has little like rungs of a ladder, little links running down between the two backbones of the DNA molecule, and there are letters shown here as A and C and G and T, and those letters encode the information that describe the proteins, plus a lot explains a lot. It specifies a lot more. So basically, there is a way of translating this information that's carried in the DNA into these machines, into these proteins here. And often there's multiple proteins, as is in the case of ATP, multiple proteins that come together to produce a single machine. And that's what's represented here. These particular three proteins come together to make a molecular machine. So we're going to look at a little bit of what's involved there, just to help you have a little better idea. I hope you're being patient here with me, but so we can understand these other 9 points a little better, and have you have no doubt in your mind that there's absolutely no way a materialistic process could lead to a living organism. Absolutely no possibility. I'm hoping. To me, that's absolutely clear. But most of us have been so programmed with this other viewpoint that there's some reprogramming that needs to happen. Okay. The proteins that unite to form cellular machines are themselves manufactured by special cellular machines known as ribosomes. And so you may not have encountered that word before, but that's what these are very important machines in the cell. And this is the ribosome, this little thing right here. And basically a strip of messenger RNA, which has copied the DNA in the cell nucleus, come out, the DNA unzips a little bit, and it's transcribed. And I'm not showing that process, but it creates a copy, an RNA copy of the DNA in the cell, a short segment of it that's for a specific protein. And so that comes out of the nucleus like a little tape. For some of you, boy, if you lived in the 60s, there were machines that read paper tape. And it's a similar kind of machine. You run this tape through it that carries the instructions for making the protein. All right. So part of this machine reads this tape. It pulls the tape through it. And then another aspect of the machine, there are out in the cell environment, there are little things called transfer RNA, which have a certain code on them and are carrying an amino acid attached to them. And so what happens is that they're around, and they get if the right one will get pulled into the machine and carried through the machine, and the machine catalyzes the reaction to hook this amino acid onto the previous one in this growing chain that forms the protein. So, and then when it gives up its amino acid, it goes back and is refreshed with a new amino acid from the cellular environment. So this is a absolutely important machine. Everything, every machine in the cell is made out of protein. So this is the machine that makes essentially all the machinery in the cell. And in the case of our bodies, we have about 100,000 different proteins in our bodies. And so this same machine, it doesn't have to be it's one machine that's able to make those 100,000 different proteins just with a different tape running through it. Okay. Just to repeat what I said in words so you can see, the ribosome is a complex molecular machine found within all living cells that joins amino acids together in the order specified by a strand of messenger RNA to fashion a protein. Ribosomes consist of two major parts. The small subunit which reads the messenger RNA, and the large subunit which joins amino acids together. And each of these subunits is composed of both RNA and proteins. So this ribosome is a little bit different from most of the other machines in our cells in that it not only is made of protein, but also has some RNA parts to it. But they're all intertwined together. And so this next picture shows the structure of the bacterial ribosome. This is the one, a little simpler one than it is in our bodies that's in bacteria. But it performs this very same function. So this part is the part that reads the tape, that reads the messenger RNA tape, and this is the part that's responsible for joining for accepting these transfer RNAs with the amino acids and forming the protein chain. Okay, and so there's now these are, I'll show it. These represent strands of either protein or RNA, and so there are thousands upon thousands of atoms represented in these little strips. So just mind-boggling complexity. And for these things to work, essentially every atom matters. The atoms need to be in the right positions. And so, again, just hard to comprehend the complexity we're talking about.
The ribosome manufactures proteins.
Yes,
But it itself is made out of protein.
that's right.
So you could almost ask, where
How
did
do they
that protein
make it?
come
Right.
from?
And that's what I'm getting to
Okay,
next.
sorry.
No, it's good. You're starting to put it together.
It's
What we're
chicken or
talking
the
about is irreducible complexity. The system will not work without its parts.
Right.
There's no way to put it together in a piecemeal fashion, like Darwin proposed. It's either all or nothing. Okay? And that's the next major point I'm going to try to drive home. It's either all or nothing. There's a Darwinian scenario simply cannot put something like this together. All right.
John, does the protein chain, does that actually make up our DNA?
No.
No.
Or you say no
The protein
DNA.
chain.
No, a DNA. No, the DNA carries the instructions, the specifications for. It carries the very special code, if you will, a very special order of amino acids that are required for these very specialized molecules that form proteins in living organisms, and so the DNA is called, it's deoxyribose
Nucleic acid. Deoxyribonucleic acid.
nucleic acid, and so that's what. Okay, and so instead of a protein, it's nucleic acid. It's a structure containing nucleic acids, And so but there are proteins that are very intimately associated with DNA that help it help it even to fold. It folds very compactly in the cell nucleus, and without these helper proteins, it could never fold up like it does. So but the DNA itself is not protein. Okay. Those are all good questions. All right, so let's take a look at what sort of, you saw those letters in the RNA. It turns out that there are 64 three-letter RNA code words that code for the 20 amino acids. So here is the vocabulary that RNA uses for the various amino acids. And here are some of this is the name of the amino acid here. You may have heard of some of them. Leucine,
Phenylalanine for those that
lysine. There are certain essential amino acids. Our bodies are made up of all these amino acids. In some cases, our bodies can synthesize the amino acids. Other cases, we have to take them in our food. So again, amino acids are crucial for, well, that's what proteins are made out of. So what you eat matters. If you eat junk food, all this cellular machinery is has got to cope with it. Anyway, perhaps it helps you understand why protein, I'll just say that, why protein is important in your diet, and why a complete protein is important. Even if you're a vegetarian, a single source of food does not give you complete protein. And this is the reason why you need all of it. You need all 20 of these amino acids. Okay. I mean, what else was going to say? Oh, okay. So another point here is that each amino acid is coded with a triplet of these genetic letters, and the DNA code. This is what's in the RNA code. And the DNA code is the same, except T replaces U in the code. And these I'll show in the next slide. This shows the molecular structure of RNA, or at least one backbone and the nucleic acid associated. This is what's called a sugar phosphate backbone here, and so this is the sugar, this is the phosphate, and connected to it, each one of these is an amino acid. Is a nucleotide, and there are 4 kinds: adenine, cytosine, guanine, and thymine. And so these form the genetic letters in DNA. And so it takes 3 genetic letters to specify a protein. So that's how these amino acid. So each amino acid in the protein is specified by 3 of these letters, 3 sequential letters in the DNA. And if you're getting overwhelmed, well, basically just trying to give you a flavor of just how complex all this is, but how incredibly well designed it is. So what happens then, the ribosome reads this tape, which is a section of transcribed, it's messenger RNA, that's a copy of a section of the DNA, and the ribosome generates this sequential chain of amino acids. And typically these are like 150 to 200, in some cases 300 amino acids in length. Okay? And the key thing about it is that each one of these amino acids has what is called a side chain with a certain charge structure on it. And those charges then can interact with other amino acids. And for a live protein, these straight chains come out and almost immediately start coiling, form a helical structure like this. So this is called an alpha helix. And then those can form tape-like structures or sheet-like structures, which then fold and then produce a three-dimensional structure, three-dimensional protein, and often several of these will come together to form a actual machine. And this is the one that's shown here is hemoglobin, which has four individual strands of protein that come together, fold together, form a very compact 3D structure that's able to carry a molecule of oxygen, for example. Anyway, so this gives you an idea of how these machines are manufactured in the cell. So here's a more specific case, and our red blood cells are simply just little sacs packed full of these hemoglobin molecules. It's a red blood cell has no nucleus, very simple, it's just a package full of hemoglobin molecules. But there are four chains, 2 alpha chains and 2 beta chains that make up the hemoglobin molecule, and at the center of each one of these is what's called a heme group. That's why it's called, or connected with hemoglobin. And this has an iron atom in it and is specially suited for carrying a oxygen molecule. Okay. So again, see just the incredible design here. And as I said, something like 100,000 different types of protein to make the machines that allow our bodies to function the way they do. So what begins as 4 linear chains of amino acids folds into the 3D hemoglobin structure. And so we get this chain folding, forming this, in some cases, an alpha helix, and other cases forming what's called a beta sheet. And then this all comes together and typically folds into a compact structure like you see here. And then these come together to form, this is called the tertiary structure. This is the quaternary structure, the 4th level structure that actually is the machine. So what I'd like to do is just to help you grasp this a little better. I actually have here the sequence of amino acids for this one. I have this one is the beta chain. I have the alpha chain in the other little bag, but this we went to Walmart and got these little links, and they didn't have 20 different colors, so we put colored tape around some of them to give us the 20 different colors. So this actually is the beta chain for human hemoglobin. Okay, and this illustrates what that ribosome produces from that messenger RNA tape. It puts these links in the exact order to allow this thing to fold up compactly so that it can carry an oxygen molecule. Okay. So anyway, I might. All right. So this And I have a little red arrow, so this is the beta chain. Each letter here represents a distinctive amino acid. And it turns out if one of these gets messed up, a mutation, this is a destructive mutation, if it occurs in this 6th position here, let's see, I can't remember, it's changing a valine into I forget what the other, what it changed, no, it's a E is, I have to use my crib sheet, I can't. Yeah.
Boy, that age is
Yeah,
really
yeah,
starting to show now.
E stands for glutamic acid. That's the amino acid. If it gets replaced by valine, the condition is known as sickle cell anemia. And so that's an example of what happens if just one of these amino acids gets changed. Okay. Now, I'm going to get to one thing. I'm going to get to next is evolution, to account for how life could arise from through random process. Somehow has to come up with this particular prescription for a protein. It has to do it by a random process. And so let's consider here how many possibilities there are. You could ask how many different variations are there in this chain? Well, at each position, each position could be filled with 20 different possibilities. So there are 20 possibilities for this site, 20 for this one, 20 for this one, 20 for this one. Okay, so the length of chain is, there are 146 units here. So the number of possible variants is 20 to the 146th power. And that number is so gigantic, there's nothing in the whole universe it could be compared to. There's estimated to be no more than 10 to the 80th atoms in the entire universe. And only about 10 to the 18th seconds in 4.5 billion years. And I mean, there's no way you could no way you could possibly run through all the combinations. No conceivable way you could ever test and come up with and find by a random process this very special structure. So I'm giving you a preview of what I'm going to talk about in a few more moments. So it's just utterly absurd to appeal to mutation to be able to identify a structure like this. And so what a mutation would be, I've got each of the 20 possible units here. If if I wanted to try to improve on this, I could pick one of these and substitute one of these in there and see what happened. But it would take, you could only test a tiny fraction of the possible combinations if you wanted to transform this into, try to make it into something else. Okay. And to me, this should be obvious to any scientifically trained person. Any scientist should easily be able to grasp the absurdity of appealing to natural selection or materialist processes to find structures like this. Okay, and I show if you take both of these together, the alpha chain and beta chain, the number of possible variants is 10 to the 373rd power here.
So John, real quick, if you can cross a horse and a donkey and get a mule, or you can derive dogs out of wolves, they're still going to have dog DNA, but you can get a Chihuahua and a Shepherd, which are going to have some variations, right? But when it comes to humans must have a totally unique, because they can't cross with anything else, correct?
Well-
And you couldn't pull.
Well, like the various kinds of dog varieties, they still have the same basic DNA.
Right.
And humans, the various what we would call ethnic groups, Chinese. Basically, it turns out that we're extremely similar genetically, just incredibly similar. The genetic differences are very small, but God built in that much variation and probably more in Adam and Eve, and so we'll look at some of those issues a little bit more later.
The example you gave were still within that particular species,
Well,
Yeah,
but a wolf to a dog,
but it's
that
still,
is a different genus, right?
No,
no,
Right?
it's still
Same
It's the
kind.
same
They're both
creation.
canine, they're both
but if you
canine.
go down to point number 8, the staggering number of transitional forms is unbelievable. So you can't cross a German Shepherd and a donkey. You know?
No, I
So
understand
that's,
that, but.
that's where the tradition.
Basically, the phrase after its kind occurs 10 times in Genesis 1, as if God is trying to make it very clear, very specific, that He's put reproductive barriers between around these created kinds. They can't cross with another kind. Now we'll look at just where in the normal taxonomy, levels of taxonomy, does what these created kinds occur, and I'll discuss this. But it appears that it's roughly, in most cases, at the genus level. So all the various related species, all the species in a given genus, have the potential of crossing and producing fertile offspring. At least hybrid. So in the case of the, well, anyway, we'll get into that topic later.
Well, crossing naturally, but what about now that we have gene splicing? Do you think we can mechanically now make all kinds of things happen?
Well, that is, that is interesting because now we have the technology to introduce genes from some other place into a species where that gene normally does not occur. So, for an example is, and I don't know the names, but there's a gene that makes plants Roundup resistant. Okay, and that gene can be transferred into crops like soybeans. And so I know the guy that developed this technology. He's a close colleague of mine. So I think of him these days when I drive through the Midwest, say Illinois, Indiana, Ohio, and look at the soybean fields, and there's not a weed to be seen. You're perfectly clean fields. When I was growing up in Texas, as a kid, one of the major tasks was as hoeing the weeds, and a cotton field free of weeds was, that's what you were striving for, but you only achieved it with a lot of back-breaking labor.
So what's the consequences in the soybeans?
And then we eat it.
You eat it. And as far as anybody can tell, that gene has no effect on humans. But some
Well, how
people
do they know that?
some people say, well, how do you know that?
Some people.
John? It's glycophosphate and it's an enzyme that's only in plants. We don't have it, but they do add, recently they've added an adjuvant to the Roundup, which makes it carry into the plant. And we are susceptible to that. It breaks down basement membranes. So
Yeah.
that's the danger, if there is any danger.
Okay, so Sharon, she's a biologist. The chemical in Roundup is glycophosphate. And so this gene makes the plant it prevents glycophosphate from damaging it. And what she's saying is that this, that glycophosphate, well, she's saying that the way of getting that into plants more efficiently, can be a problem because it gets,
It affects basement membranes.
yeah, it can affect our own.
So that's why we get more wrinkles now, is that it?
So,
I vote for that.
so anyway, that's a.
Son does it.
So as humans have been able to understand what we're talking about, it's given them ideas about, well, how can I apply? Is there some way I can make a buck out of this?
Bottom line.
Okay, I really want to get through this. So how does the Neo-Darwinian theory explain how a new protein with new functionality arises? Well, as I've said, that theory, it relies on random mutation of existing genetic instructions to generate the new DNA specifications. Crucial to whether the protein functions or not is whether it folds correctly, as I've already said. And so I would just want to go into a little more detail about the issue of protein folding. As I said, it can provide an alpha helix and several what's called a beta strand, and several of the beta strands can form what's called a beta sheet that looks something like this. And so this is the way that the basic linear protein, two ways it can fold. It can coil and begin to fold. But then a crucial step is what is called a protein fold, where these beta sheets basically can fold over to produce a three-dimensional structure. So we're some terminology of primary structure is the linear chain. That's as manufactured by the ribosome. Secondary structure, successive portions of linear chain form either alpha helix or a beta sheet. Tertiary structure, alpha helices and beta sheets collapse to form a protein fold. And so all of these structures are governed by the charges that exist on the side chains of the amino acids. And so it's very much of an electrical phenomenon how these things are able to fold. And so the proteins that fold are extremely unlikely. And one guy, this guy is actually a creationist, has worked on, published in 2004, some work involving what's called site-directed mutagenesis experiments, where today you can go in and selectively change the DNA to code for a specific, change one amino acid at a time. So he did that and looked at the effect on the resulting protein to fold. And in particular, this beta lactamase enzyme, which is a protein. It's an enzyme, but it's a protein. And found that the ratio of the stable folds with function to the whole set of possible amino acid sequences of that 150 length was 1 in 10 to the 77th power. So these getting a protein to fold is just incredibly unlikely. And so again, just to say what I just said, how many functional folds exist among all the possible combinations? And so the number of possible combinations, if you've got a chain of 150 amino acids, is 20 to the 150th power. And how many have are 10 to the 195th power. And how many of them, what is the fraction of those that produce a functional fold? Well, it's this fraction of that. So it's 1 in 10 to the 77th power.
So that is an additional variation from the previous slide you had about
The variations of amino acids.
Yeah, this is, we're saying that it's conceivable out of that gigantic number I put up for, say, hemoglobin, that there might be multiple amino acids that could still give some functionality, still fold and give functionality. So we're looking at what, allowing for all those functional possibilities. What is the ratio of the functional possibilities to those that have no function at all? So in other words, really to make this case strong, we need this kind of study. We need this kind of information. All right. So now we have the likelihood of getting a functional fold. So if we've got this trial and error process, which is what all that Neo-Darwinism has, the issue comes up: how many total tries do I have? If it's a trial and error process, how many tries do I have? It's like winning the lottery. How many lottery tickets can I buy? That determines whether I have any significant chance or not to win. And so the number of trials that Neo-Darwinism has can be no more than the total number of organisms that have ever lived in the history of the earth, assuming they're all genetically different. That's the maximum number of trials. So we're now going to try to come up with another number. How many organisms have ever lived on the earth?
Oh my.
And so what we're going to do is we're going to focus on one-celled organisms, bacteria, which can reproduce rapidly, and in today's world there are a lot of them. So there's been a study made on how many single-cell organisms are currently alive on the earth, and what is their reproduction rate. All right, so this guy estimate the reproduction rate of all single-cell organisms on the earth. And that includes all the bacteria and archaea, which is like a bacteria. These tend to live in extreme environments, extremely hot environments, like the hot springs at Yellowstone. Okay, and most of these organisms, it turn out, live in the ocean, and actually live in the top part of the ocean. So we have a huge number of single-cell bacteria-like animals that live in the ocean. His number came up to be the number of how often they reproduce, 1.7 times 10 to the 30th organisms, single-cell organisms per year. Okay? And just to give you an idea of how much biomass that is, they estimate that these organisms constitute somewhere between 60 and 100% of the estimated total carbon in plants, plants that we can see on the earth, all the forests and trees and things. So that that's a considerable, fairly large amount of biomass involved in these single-cell organisms. But that's the number we're going to use. Actually, I'm going to increase this a factor of 10 to account for earlier times in Earth history where there might have been more life, and also to account for all the other organisms that are not bacteria, which are a tiny fraction of them. Okay. So let's multiply that rate by 10 to account for all other forms of life and for conditions more favorable for life in the past relative to today. Let's assume at least one mutation per individual so that all organisms are genetically different. And let us, to be generous, give the evolutionists slack and let's use their estimate for the age of the Earth, 4.6 billion years. So we multiply 10 times that number from that paper times 4.6 times 10 to the 9th years, and we come up with not quite 10 to the 41st power number of organisms that could have possibly ever lived on Earth. Okay? So we have that as pretty much an upper limit of how many trials, how many rolls of the die have occurred in Earth history. That's how many trials the neo-Darwinist has to get not just one protein, but everything, the evolution of all life. That's all the trials they have to work with. Okay? So this limit on the number of genetically distinct organisms that could have ever existed is a generous upper limit on the total number of trials available for all neo-Darwinian trial and error search attempts combined, not for merely a single new protein fold. And for some of you, that number is 1 followed by 41 zeros. That's okay. It's still a big number.
Our national debt.
Yeah,
You know, our national debt hardly comes anywhere close to that. Our national debt's nothing compared to that number.
We got a ways to go.
Yeah.
Plenty of money.
So that means quantitative easing. We've got, you know, a lot more. You know.
Yeah.
Okay, but the probability of finding only one functional protein fold in an amino acid sequence of only 150 units is only 1 in 10 to the 77. With at most this many tries available, the odds of succeeding are only 1 in 10 to the 36th power. That's 1 chance in a trillion. So the national debt's only like 19 trillion. So this number is tiny, but that number is tiny compared to this one. Okay.
So you're saying there's a chance.
I would say it's a less than a snowball's chance. Okay. So again, I emphasize this is just one fold in one protein out of the 100,000 different proteins we have in our bodies. The one has at most 10 to the 41st tries for all of them combined, plus all the others in all the other organisms. Okay. This number of trials is available so pitifully small relative to the number of molecular configurations that are possible. A trial and error process is guaranteed to fail even for one live protein. Okay, there's just simply not enough trials, not enough rolls of the dice. And therefore, I say it's utterly inexcusable for anyone, and certainly a scientist, to claim that trial and error processes can discover the blueprint for even one biomolecule, much less 100,000 of them. These molecules are incredibly special. No material process can possibly arrive at their design. This issue, the lack of enough trials, is alone sufficient to falsify the entire neo-Darwinian framework. That framework no longer deserves status even as a scientific idea. It's utterly ridiculous, utterly absurd, and yet it's what's taught in Ramona grade school, middle school, high school, and what our children are being indoctrinated with, right here in Ramona. Okay. And
That's
so
why he's here.
Huh?
That's why he's here.
Okay, and unfortunately, I'd hope to get through this section about what the meaning of irreducible complexity. I've already basically explained it. It's the fact that, well, let me just introduce it here. I've got a few seconds. Darwin acknowledged that his theory of gradual change by natural selection carried with it a stringent challenge. He admits that in Origin of Species. Darwin states, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down.
Well, hello.
And I would say that every one of these proteins, every single one in the body, every different type, is irreducibly complex. Plus the machines that these proteins form are irreducibly complex. Plus the biological pathways that multiple machines are needed for are irreducibly complex. Our cells are irreducibly, each cell is irreducibly complex. Let me describe what, first of all, what that means. Michael Behe in his book, Darwin's Black Box, asked what type of biological system could not be formed by numerous successive slight modifications. His answer, well, for starters, a system that is irreducibly complex. By irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. And to illustrate that, Behe uses the common mouse trap. Remove any one of these 5 parts and its functionality is completely destroyed. It will not catch a mouse. You take away the spring, take away the hammer, take away the holding bar, take away the catch, take away the platform, and you do not have a functioning mousetrap. All the parts have to be there for that mousetrap to have function. Okay? And so I think I have to stop here. Let me just see what thousands upon thousands. I would say hundreds of thousands of features and subsystems within living systems are composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system effectively to cease functioning. Okay, so I think you're starting to get the picture that neo-Darwinism has got huge problems. And so I'll finish, Lord willing, I'll finish up next week on this point, and we'll start more quickly going through the others. Next week, I want to especially cover the idea that these specifications in the DNA represent a non-material phenomenon, non-material reality, and that by itself falsifies materialism, philosophical materialism, which we're taught is that's a presupposition that just pervades our school system, that there is no reality that's non-material. Well, I'll show next week, Lord willing, that there's reality all around us that we participate in almost every waking moment, essentially, of our lives, that can be shown is non-material. And anyway, come back, invite your friends, and Lord willing, we'll have another good session next week. So let me close in prayer. Gracious Father, thank you for your truth that sets us free. Thank you that your word, the Bible, is the Word of God, is truth from start to finish, and that we can rely upon it. Thank you for the light you're giving in the day in which we live, wherein we can be very confident as we share your truth, as we stand upon your word, and I pray that you'll help all of us grow in that regard. Thank you for this opportunity to consider these things. Give you praise, give you glory. In Jesus' name. Amen.